I'm going to share with you our team's results on Lassa fever in West Africa. Lassa fever is a severe hemorrhagic fever caused by the Lassa virus, which was identified for the first time in 1969 in the Nigerian town of Lassa. Lassa fever follows exposure to infected rodents, but it can also be transmitted from infected persons, and particularly in medical settings. In 2022, MSF treated 79 severe cases, mainly in Nigeria, and there's a high case fatality rate because patients are seen at a late stage. In the literature, the CFR is usually between 15 and 20 in hospital patients and up to 30% in pregnant women. Currently, the standard treatment is ribavirin, and there are no preventive drugs or vaccines. As you can see on this map, Lassa fever is endemic in much of West Africa, where the CDC estimates that it may be responsible for 100 to 300,000 cases and 5,000 deaths per year. It is a disease with a pandemic potential, and a study published last year showed that more and more people in Central and East Africa are going to be at risk over the coming decade, and particularly because of the climate change, which is likely to increase the habitat of that is suitable for these rodents. Now, for all these reasons, Lassa fever has been placed on the WHO list of priority pathogens in order to speed up the development of a safe and effective vaccine. Now, of course, developing a vaccine is a long and costly process. Currently, there are several candidate vaccines. They've completed the preclinical phase and are undergoing phase 1 and 2 to assess their safety and efficacy. Phase 3 are very important in order to assess the efficacy of the vaccines and, of course, condition their regulatory approval. However, for Lassa fever, we still lack some crucial information to design an optimum phase 3 trial. Firstly, we don't have a clue about the incidence in the population. We don't know how frequent it is. Secondly, we don't know exactly what is the level of protection that is conferred by prior exposure to the virus. Thirdly, the groups that are the most at risk haven't been clearly identified. Finally, we don't really understand the spectrum of severity. And if we can't answer these questions, well, it's likely that a phase 3 trial may not be well designed, therefore making it impossible to assess the efficacy of the various candidate vaccines. And this is what led to the implementation of the research program on Lassa fever in order to have a better understanding of the epidemiology and so as to design phase 3. Enable is an international consortium funded by CEPI. Epicentre is in charge of data management and statistical analysis. It covers 7 sites in 5 endemic countries: Guinea, Sierra Leone, Liberia, Benin, and Nigeria. At each site, the study is conducted by a principal investigator with a dedicated team and lab. Now, the main objective of the Enable study is to assess the incidence rate of Lassa fever and also the incidence of the virus, which are 2 different things. To do this, we recruited 20,000 participants aged over 2 years in the 5 countries. Those participants were sampled, for their blood was taken so that we could determine their exposure, prior exposure to the virus. Then we got in touch with them by telephone every 2 weeks for 2 years to find out if they had a fever or symptoms. Those with acute illness were referred to health center for treatment and PCR testing. If the results were returned positive, they were isolated and also notified as symptomatic cases of Lassa fever. So I'll leave you to imagine the workload behind all that. In addition, we recruited 7,000 participants, including 4,000 among these 20,000 who agreed to provide a blood sample every 6 months so that we could carry out, we could test their serum and determine whether they had been exposed and infected by the virus. The study started in December 2020 in 3 sites in Nigeria, followed by the 4 other sites in the second half of 2021. It's going to be complete by the end of this year, and the final results will be available by mid-2024. Let me share with you the interim results based on the analysis carried out in October 2022. So I'm going to illustrate our results while focusing on our results in the Edo site in Nigeria. This is where we have the longest follow-up and also serological data available at inclusion at 6 months and 12 months. In this site, with some 5,000 people, we identified 43% of participants exposed to the virus, confirming that the zones are highly endemic. Plus, there is also an incidence rate of infection with the Lassa virus of 13% per year, which means that 1 person out of 10 is infected by the virus in these communities. Conversely, there were only 18 Lassa fever cases among 5,000 participants, which gives us an incidence of Lassa fever of only 0.2% per year. In other words, just 1 infection out of 65 people can lead to Lassa fever. So this interim result shows us that the infections are frequent, but that the fever, even in endemic zones, is rare. So on this basis, we wondered whether these frequent infections could give people some sort of protection against the disease. To answer the question, we compared the incidence rate of people with antibodies and those without. And though the difference isn't statistically significant because of the size of the sample, it leads us to think that there could be a protection of approximately 60%, and 15% of positive participants had a seroconversion every year. So the pre-exposure to the virus could temporarily reduce the risk of becoming ill with Lassa fever. We also tried to compare our data for children and adults. Our results show a significantly lower seroprevalence in children, and conversely, a higher incidence of infection and disease. These last 2 trends are not statistically significant because of the small size of our sample. However, we can assume that children are at least or as much at risk as adults because of their low pre-exposure to the virus.
So these were the epidemiological results, but what's the implication of those interim results to design a phase 3 vaccine efficacy trial? This is an individual randomized trial. Participants are given either a Lassa fever vaccine or placebo vaccine. The main objective is to measure the incidence in both groups and compare them to measure the efficacy of the vaccine. To design such a trial, we have to make an assumption on the real efficacy of the vaccine. Here, we assumed that the true efficacy was 70%, slightly above the 60% natural. And also, we had to make an assumption on the incidence rate of Lassa fever in the placebo group. We use enable results and take the estimate of 0.2% per year. Given that low incidence rate, the trial should enroll 20,000 participants to measure the efficacy of the vaccine with enough precision, and to increase the precision by a factor of 2, we have to multiply by 4 the size of the sample, which, of course, from a practical standpoint, is problematic. So the final decision on the size of the sample will be a compromise between precision and feasibility. Another key step in designing a phase 3 trial is the population to be studied. Often children are excluded from the study. It was the case with the Ebola vaccine in Guinea. Some of our interim results show that the children should be enrolled in the study because they are at high risk of contracting the disease. They could contribute to help us measure the efficacy of the vaccine and be included in the target groups once the vaccine is fully approved. First, we have to check that it's safe in children, and that's what we'll do, doing our safety and tests. So, ENABLE is the first epidemiological study aimed at measuring the incidence of Lassa fever disease and LASV infection in West Africa. It will use it to inform the design of phase 3 vaccine trials. A large sample size is needed and children should be included. Those interim results are based on a partial data from a single site, but this will change when we include the other sites and all the follow-up. Those results concern highly endemic zones and cannot be extrapolated to all of West Africa. During the next 3 years, Epicenter will still be involved. We continue being involved in research on a vaccine against Lassa fever. ENABLE results will be available mid-24. We take part in a phase 1 and 2 trial on safety and immunogenicity, and this feasibility on a larger scale phase 3 trial. We're sure that eventually in the 4, 5, 6 years after that study, we'll manage to develop a vaccine. I'd like to thank all the partners of ENABLE program and the steering committee that allowed me to share with you those interim results. Thank you for your attention.